I really want to talk about meetings today because, in a way, that's what we're here for. I really want to talk about a little bit about the founding of what has been called the American Phage Group, which was really the group that first started having phage meetings. But I think that really we ought to start talking a little bit, because I'm the only one probably who's going to indulge you with this. As I say, I'm one of these elders, wise I don't know about, but anyhow, we've heard about these two guys here, and there are two biographies out there. One about Peter Tort, written by his son Anthony. I think it's appropriate, the title that he gave it, In Focus, Out of Step. And the other is a biography, a scientific biography of Felix Thorel. And as Forrest indicated, there's some controversy over whether you should, when discovery took place. Philosophers of science have argued for a long time about what discovery really means and whether it's an individual event or not. If you look at Tort's paper, which is called something like the Studies on Ultramicroscopical Viruses, he was referring to vaccinia and distemper of dogs, not to phage. He didn't. Don't mistake that paper for being about phage. It was about vaccinia and trying to grow vaccinia in cell-free medium. He incidentally noticed the stuff he called glassy transformation. Thorel, however, was the one who conceptualized phage and actually convinced other people as to what was going on. As Thomas Kuhn said, you can't really say the first guy who put a cork in an empty bottle had discovered oxygen. And I think, we can talk about this at the coffee break, but I don't want to belabor that, but it's a controversial subject. So you can think about the sort of trajectory of phage research. I'm not supposed to wander off this spot, I guess. In the beginning here, and if you look at it, it really did start out with an interest in phage therapy. These were thought to be microbes that infected other microbes, and they could be useful. This is pre-antibiotics, and so there was a real interest and need for this. It was only in the late 30s that phage became a biological phenomenon, and they became the model for reproduction. Now, the word was reproduction, not replication, because this was like rabbits. It was more like how does one beget like, not how does a molecule make another copy of itself. And indeed, because viruses were thought to be proteins, and the problem of protein synthesis was a total mystery, the early phage workers really started to study phage because they wanted to understand protein synthesis. And the main method that they used was radiation biology. This is a forgotten era because we now have tools such as isotopes and other tools to study these things. But before World War II, when one had availability of isotopes, bombarding cells and particles with radiation was the way one did it. This is what the physicists had done with the structure of the nucleus of the atom. Why wouldn't it work for cells and viruses and proteins and everything else you couldn't see, but you had to study? And so there's a whole strain of radiation biology. As a matter of fact, Jim Watson's PhD thesis was on the radiobiology of phage T7. A whole lot of goddamn survival curves, and changing the media in which you do the irradiation, and something called the histidine effect, and so on. Radiation was really a major tool. Now, here's a sort of early engram of phage literature, just to show you the various trajectories here. It's that one, okay. So this is by a German named Hans-Jürgen Rettig, published in 1956. A very handy little book to have if you can find it. And he looked at the phage literature, and of course it starts in the pre-20s. And can you get that out of there? Yeah. Very good, thank you. And what it notices is there's sort of a fall off from the 1940s. This is, of course, partly due to the war years, but it's partly due to the fact that this is the antibiotic era, and interest in phage therapy was dying off. But it was in the 45 to 50 area that research took off again. And it's this area, that time period, that I want to talk a little bit about. So phages, however, were of interest to the humanists before this. And for example, I have a little example. There was a famous book called Arrowsmith, written by Sinclair Lewis, for which he got the Nobel Prize for Literature, the first American Nobel Prize for Literature. And here's a movie clip of an Academy Award nominated movie from 1931. This is young Dr. Arrowsmith.
2 nights, couple of weeks, I don't know. Look. No, we should look at it, Chief. Yeah. Think I may have found something. What? Don't ask me. All I know is it's hell on the banks. Dissolves them. Eats them up. Slaughters them. Wipes them off the... Oh, you're right. What's all right, Terry? I fancy honestly all in when I started. Oh, and food. Need a drink. Oh, Terry, need that one. I'll get it for you.
That's the best part.
Big thing here. A great thing. Can you look? Now. No. You must find out what it is. How many different kinds of bacteria it will destroy. That way you will find out what it is good for. You will begin working in earnest. I am very glad, Martin. I'm glad you feel that way, sir. Boy, you're shooting hard. We're going to celebrate. I don't care how tired you are, we're going to celebrate. I always knew that, kid.
So here were the headlines. The phage were discovered at the McGurk Institute, which is really the Rockefeller Institute. And it made quite a splash, as you can see here. And however, it was not all joy in Mudville.
Of the paper I just signed. Yes, Dr. Gottlieb. We're busy at the moment. Yeah. Busy turning my friend Martin into a megaphone. Oh, that's all right, Chief. Something has happened. Or is something not altogether bad. Martin is not the discoverer we thought he was. But you don't mean that someone has stolen the mark on us? A Frenchman. Derrel of the Pasteur Institute of Paris. He has published his report. It is the same thing. You have got into the newspapers too soon, Dr. Tubbs. Of course, there can be no talk of head of department now. No raise of salary either. Well, I've got to say something to those reporters.
So nothing has changed, right? However, here are the real headlines. This is very parallel. This is actually true. This is not fiction. This is Emory Ellis working at Caltech in 1938. New battle won in war on diseases, for example. And look at the rhetoric here. With the aid of a microscope, the scientist actually witnesses carnage and military victories on a scale never approached by human soldiers. An unseen virus now thought to consist of protein molecule attacks one of a group of bacteria. This was in the LA Times, 1938. And this is Emory Ellis, who I'll talk about in a few minutes, looking at this carnage under the microscope. Ellis was the researcher at Caltech that Max Delbrück came to work with when he came as a postdoc to the biology department at Caltech. And Ellis and he worked together on a couple of the very early phage papers. Ellis then ended up, because of a dispute with his sponsor of his fellowship during the war, ended up changing his career. He became famous as the inventor of the Sidewinder missile and became the head weapons analyst at the Pentagon, and had another career totally outside of phage. He's quite an amazing guy. Now, as I said, about this time, the real question that biologists were interested in was how phages reproduced. And the question, were they synthesized from cellular components, or what was actually happened to the phage material? Did it get passed through or not? And so it was obvious there was genetic transfer, but there was a real question as to whether there was physical transfer of parent to progeny. And so this whole business of parental to progeny transfer of substance as well as information, it was not called information at that point, it was called genetic or hereditary material. This became the central focus of a lot of research. And so here's the sort of canonical history that you find in textbooks. I'm not going to dwell on it too long, but it's, as I say, it's the sort of stuff almost anyone could come up with. However, I think it's important historically to unpack this a little bit to see what really was going on. And so these are some of the real experiments that people have heard about. So for example, the question of what is the source of the material was a project undertaken by people trying to understand protein synthesis. And Seymour Cohen was probably the first one to do some experiments along this line. And indeed, this line of research was taken up by Frank Putnam and Lloyd Kozloff, and by Angus Graham and his colleagues in Canada first, to find out where does the parental DNA go and what does it become. And of course, only then did Hershey and Chase do their experiment of actually labeling, when they got a hold of label, labeling experiment. So it wasn't just grew out of the brain of Al Hershey to do this experiment. It was, like most other experiments, built on previous foundations. And I think if you read Lloyd Kozloff's PhD thesis at Chicago, you find that a lot of the ideas that Hershey then developed more in detail were already there. The question of how much parental DNA persists was a real problem. One of the amazing things was when you do a parental to progeny transfer experiment that'll label the phage DNA and see how much ends up in the progeny, there seemed to be a 50% limit. Nobody could get more than 50% transfer. And indeed, Jim Watson entertained the notion that only one of the two DNA strands was passed on. And indeed, people thought that this was, Gunther Stent, for example, had a whole paper explaining how this was really the way DNA replicated. That you threw away one strand and you kept the other strand, based on this parent to progeny transfer. Later on, when the rolling circle model came along of Wally Gilbert and David Dressler, again, this suggested that one of the strands was conserved and the other strand was made new completely. And it was only really when Esselstyn and Stahl did their experiments with phage that it became clear that both strands were transferred and that probably the loss, this 50% threshold, seemed to be a technical problem. But for a long while, this was a debated question and confused people's models about how phages actually reproduced. So, as I said, this was a problem to study protein synthesis. We have data, for example, from the phage course, from the phage meeting notes of some of the earlier scientists. Fortunately, they've saved them. And I hope this kind of stuff will be saved for future historians, not just digitized, but actually the physical stuff. So, as I said, one of the things that came along that helped a lot was radioactive tracers that helped to test for material continuity. So, these were the kind of problems that were set for phage workers, but how did a problem get set? Who determined what was going to be studied and what was not? Who determined what was an important question? Well, this is where the social structure of science becomes important. And so I want to really turn to the question of how did a phage group form and how did it become a research community? What were the strategies that were formed, were used to build this group? Were there leaders and followers? How did various social and intellectual personal factors come into play? So one can think about different models of specialty development, and this is what sociologists of science spend their life doing. You guys probably don't talk to those folks, but since I've become elder, I spend more time talking to them these days. And indeed, for example, Gunther Stent described what he called the romantic period, the dogmatic period, and the academic period of phage research. A sociologist named Nick Mullins, who wrote a famous paper in 1972, talked about paradigm development, problem success, and puzzle solving as sort of these same equivalents. Now, the kinds of things that Mullins used to analyze the phage group was who communicated with whom, who co-authored with whom, who were colleagues, who were students and teachers. These were the kind of relationships that one could analyze. And so this just shows you the kind of schemes that Mullins put in his paper. He said, well, we started out with a few researchers who defined the paradigm. Then we began to have networks of, for example, colleagueship and co-authorship and so on. And eventually you develop a big enough network that it becomes what you might call a specialty. And I think that this is a little too restrictive in a way, and I have felt that it's better captured in the case of molecular biology and phage research by a model developed for computer science, actually, and cognitive science by Barbara von Eckert. And she talks more about what the group does. It defines who's in and who's out. I've asked a number of the old wise elders. Some of them may be in this room and want to comment, but several people told me, well, the phage group was defined by whether you sent your preprints to Max Delbrück for his approval before you submitted them to the journal. Elio is nodding, right? And indeed, the people at the University of Chicago, Lloyd Kozlov, told me they didn't. They were not in the phage group. Seymour Cohen, who was a biochemist, told me he was never in the phage group because apparently he didn't do this. He went to all their meetings and he was on all their mailing lists, but he was not in the group, right? I mean, this in and out sort of thing is really interesting. Who agreed on methods and standards? Well, as probably many of you know, there was a standard textbook or manual book called Bacteriophages written by Mark Adams in '59, which grew out of the phage courses, which was designed to standardize methods. And standardize, as you all know, the T phage as the group of phages to work on. Josh Lederberg and Esther Lederberg told me that they had a lot of trouble trying to introduce lambda as a phage because it was a lysogen. And the phage group itself did not really want to deal with lysogeny. Again, we may hear some more about that from Alan. But so there was an agreement on what were the key problems to solve. And if you actually go back and look at the literature, you wonder why were they trying to solve these problems? For example, there was a huge research push on something known as the tryptophan effect. The tryptophan effect was the fact that apparently T4 requires tryptophan in the medium to absorb and grow. And Max Delbrück thought that if we could only understand why tryptophan was required, we'd unlock some great secret of phage research. To this day, I'm not sure anybody knows what the basis of the tryptophan effect is. Maybe Patty can illuminate us. But the point is that
it's only T4B and not T4D.
Yeah, right.
The tail fibers now.
So anyhow, there was an agreement and setting the agenda, so to speak. And then there was the question who polices the whole group, enforcing standards and so on. And we'll talk a little bit about that. So who were the leaders? Well, you all know, I've heard of Max Delbrück. Max was an interesting character in many ways. He was in many ways thought to be the leader of this group, and he had started, when he was a physics postdoc in Berlin, having small groups in his house, which he called a little club, which started out as theoretical physics, then it brought in genetics and biochemists and photosynthesis physiologists. And this sort of journal club, if you will, in a way, when he came to America, he started to propagate this notion of basically inviting people he found compatible into his little group of people he discussed things with. And it turns out in the US, he found kindred spirits in Al Hershey and Salva Luria having to do with phage research. And they met first in St. Louis, where Al Hershey was working, to discuss phage. This was the time when hardly anybody was interested in phage. It was a very small group of people, working in rather disparate ways. Hershey was an immunologist who was interested in phage as immunogens. Luria was a radiation biologist who was interested in radiation effects and using phage as a model system. Hershey was a protege of an early phage worker named Jacques Bronfenbrenner, who was chair of microbiology at WashU, and had a rather fraught relationship, apparently, with Bronfenbrenner. Luria was a protege of physicists Fermi and a polymath named Fernand Holweck, and a phage biologist in Paris, Eugene Wohlmann. And he came to America, was trying to find a job, and eventually got a job at Indiana. Fortunately, these people left a lot of print behind them, and we can go to the archives and find out what they said. For example, Delbrück wrote to Hershey in 1943, my guess is that there will be a great rush on phage work after the war, so we better settle the elementary problems now that we may speak with authority later. So this is the kind of discipline building. These guys were already planning how to do this. Delbrück once told me that he tried to schedule the phage meetings to be the same time as the Genetic Society of America, so you had to choose your loyalties. He didn't want phage biologists dabbling with those Drosophila geneticists. You had to, be one or the other. He deliberately set out to build a set of disciples who followed his standards for phage research. So you probably all heard about the famous phage course at Cold Spring Harbor, and the very first one from 1945 I think is interesting. Here's the preamble as to what's going on, but he said the prerequisites: facility in the process of multiplication and division of large numbers, elements of calculus, properties of exponential functions. An admission test on these subjects will be given. I think those of us who knew Max realized that this was sort of his way of trying to be funny, but not always too successful. As you can see, this was an attempt to try to define and police what the group was all about. So he's talking about the course offering, therefore worthwhile and proper to offer the laboratory a short but intensive laboratory course in the techniques and problems of this field. Well, who's going to decide the techniques and the problems? You have to make a decision. And unfortunately, we have not only the course notes from some of these people who attended these early courses, but we also have the syllabi from the lab and the kind of experiments they did. So basically, this was a recruitment, indoctrination, standardization process. Mero Demeritz, the director of Cold Spring Harbor, actually got money for this course, a whole $800 from the government, from the National Research Council, because of the need for training scientists in special research techniques. So here's just a list for you of the early phage meetings. They were at Nashville, where Delbrück had gone. It turns out at that time and during the war, it was important for enemy aliens, of which Delbrück, being a German, was, to not be allowed near the coasts. There was worry that people like Delbrück and Luria, who were technically at war with the U.S. during the war, not be at the coast where they could signal submarines in from their compatriots, supposedly. From what we know about these individuals, there was no possibility of this, but this was government policy, which is why they ended up at Indiana and Nashville, and probably benefited the universities in the Midwest to a great extent. So, as you can see here, Cold Spring Harbor, of course, played a key role in hosting a number of these meetings and became the summer camp for phage biologists, if you will, for a number of years. So here's the first phage meeting agenda, and you can see the kind of topics they were interested in. Cofactor requirements of T4, 2 papers on that. Radiation biology by Maxelli, who was a radiation biologist, another radiation experiment. We got Hershey's R gene complex, calcium effects. Again, these kind of things that you wouldn't particularly think was going to be great, future potential, but this is what they thought was important. And Jacques Monod sent a letter in French that Delbrück read on adaptive enzyme synthesis. So how did they agree on the problems to solve? Well, one thing was to send out a newsletter. And Delbrück started this in parallel to what the Drosophila geneticists had done by having something called the Phage Information Service. And again, we have all these newsletters that were sent out. And so this was a description of the phage meeting at Vanderbilt in 1947. So there's the abstracts here, license inhibition by Gus Dorman. Here are the people who he sent it out to. And you can recognize the names here of key phage people. Delbrück actually kept a note on each of these copies that are in the Caltech archives as to who was on the mailing list. And one of the interesting things is to see who came and went from the mailing list. If you somehow didn't belong to the group, you got scratched off. And I have a spreadsheet that I made from all these to see how people fell in and out of favor. One of the things to do was to standardize the organisms, to standardize the methods, and as I said, this informal review system. And interestingly, one of my colleagues, Larry Holmes, had written about the Mendelssohn experiment, but in the preface to that book, he went through all 8 drafts of the the Watson-Crick DNA paper that they had sent to Delbrück. And in the course of about 6 weeks, they had gone through 8 drafts that Delbrück edited and sent back to Watson. I mean, there must have been a tremendous bunch of airmail going back and forth because Watson was kissing the ring, if you will, and was not about to submit a paper that Delbrück had serious objections to. I think they finally submitted it, even with his serious objections, because he had a problem with what he called the untwiddling problem. That is, how did the strands come undone? But this shows the extent to which Delbrück sort of was the gatekeeper, in a way, of publication. This just shows you, again, another Phage Information Service, number 10, from 1956, all right? You see here, some poor guy didn't make it, right? And again, you recognize the sort of extent of publicity here. We got, Ray Owen, who was head of biology at Caltech, Letterberg, Leo Szilard, Brenner, Crick, Ruth Sager, Elliot Volkan, all of our friends here. But again, I think, here we have a mailing list, semi-annual mailing list and so on. Well, as I say, we see how this group started as a small nucleus of a few people who were, and their students who were meeting to talk about things of common interest, to a really well-formed, organized, disciplined group of people, which we're the descendants of today, I think. This just shows the fully formed phage community by 1954. This is a phage meeting at Cold Spring Harbor. Some of you recognize the place. Some of you recognize the people, Crick, Stent, Hershey, Some of you may even be there. This is my professor, who was a postdoc there, Václav Przewalski. And so by then, this had been a full-fledged discipline and research community. And of course, we now have a whole new outlook as we start the next century. So I'd be happy to take a few questions now or discuss this later during coffee or whenever. Thank you. Do we have time scheduled for questions?
Yes.
Oh, okay.
So I'm curious if you would expand a little bit on the influence that the Phage group had on other science going on about the same time.
Yeah, the question was, would I expand on or could I expand on impact of phage on other disciplines? I must say that I think it had relatively little. If you look, for example, in 1951 or 2, I went through the Genetic Society of America. There was not even a mention of DNA at the Genetic Society of America, let alone phage. There was a group of people who were still interested in phage therapy, but they didn't seem to talk to these folks. It seemed to be quite a different, the basic science versus the applications of phage. Of course, as you know, phage typing was a development at this time, but that, again, seemed to be in the clinical microbiology area with Craigie and his people. They didn't take the phage course, for example. The phage course was taken by biochemists, who were interested in protein synthesis, by physicists who were trying to dabble in biology and wondered what's going on here. It was, I think it was sort of a separate world. Now I'm going to be corrected.
You may want to comment on the fact that the phage group had an immense influence on bacterial genetics. So the people who worked in bacterial genetics at the time were in bed with the phage people.
Yes, yeah. The phage, one of the things that Hell noticed right away in his phage therapy was that there were these things called secondary cultures. So that when you put phage, and we all see this, you put phage with bacteria that lyses, and then you let it sit around for a while, some stuff grows out. And the question is, these were resistant to the phage, right? They were resistant sometimes because they were lysogens, they were resistant sometimes because they were surface mutants, whatever, they were resistant. And this problem of resistance was plaguing the phage therapists. On the other hand, this became a tool that Luria and Delbrück used to study in their famous fluctuation test for studying mutation rates in the absence of the selective agent. And also, Esther Lederberg and Josh Lederberg used this notion of phage resistance in their replica plating test technique to explain, to separate the notion of the inducing agent or the selective agent from the mutation to the inducing agent of the mutation. It was a really crucial step in understanding how genetics and mutation work. And as I say, as Elio pointed out, this was very important in understanding genetics at a fundamental level.
So who was the most famous person to get kicked out of the phage group?
Well, first of all, that asked me to make a decision about famous.
Oh, okay.
And second of all, it came and went. I mean, that was the thing. And so, yeah.
So you just fell out of favor.
Yeah. Well, you know, we don't really know why they were taken off the list, of course. It might be just they moved and he didn't have an address, for all we know, or something. But, one of the interesting things in Mullen's work is that it shows that some of you may hobnob with a humanist, but one of the things that historians accuse scientists of is being what they call Whiggish. That is, you look at the point of view of science from the point of view of the winners. And that's really not fair because we have the benefit of hindsight. The problem is that historians who are not Whiggish don't often know the inside stuff. So, for example, Mullins was totally mystified by a guy named Raymond Laderget. Raymond Laderget, he could not figure out where he belonged because he was looking at the American phage group. Well, of course, as you know, Laderget was probably the most famous French phage biologist of his generation, and a radiation biologist. And he just didn't know, because he didn't know the interior thing. He didn't understand what happened to Marty Chase. Marty Chase got married and changed her name and disappeared from the record as Marty Chase, but she was still around. And knowing who got married, who got divorced, all this kind of stuff was important. And so some sort of internal knowledge is often helpful to the historians. But then you've got to be careful about being Whiggish. But it is sort of an interesting story to know the gossip, too, which is good. If anybody's got gossip, I want to know it. Okay.